Imagine you are a fish at 1,000 meters depth. Simple question. What do you see? Nothing. Not because your eyes do not work, not because something is wrong with them, but because there is no light for them to work with. At 200 meters below the surface, the last fraction of sunlight disappears completely. Below that point, there is no dawn, no dim glow, no seasonal variation, just permanent, absolute, total darkness. Your eyes are structurally perfect. Every rod, every cone, every connecting neuron wired exactly right, and none of it matters at all. Perfect vision in a place with no light is not an ability. It is not even a neutral feature. It is dead weight you are paying to carry every single day of your life. Here is what makes that situation more than just unfortunate. Having perfect eyes in permanent darkness is functionally identical to having no eyes. The outcome is the same. You see nothing. But the two situations are not equal because eyes are not free. Building them during development, maintaining them throughout life, and feeding the retinal tissue that demands more oxygen per gram than even the brain, all of that carries a real daily caloric price. In an environment where finding food is already among the hardest problems an organism faces, that price is not trivial. Evolution does not evaluate organs based on what they could do under different conditions. It evaluates them based on what they return in the environment that actually exists. Subscribe and stay here, because what evolution did next is not what most people expect. To understand the response, you first need to understand the cost. The retina is the most metabolically active tissue in the vertebrate body. It consumes more oxygen per gram than any other organ, the brain included. For a fish living in well-lit, food-rich water near the surface, that cost is a reasonable investment. Eyes allow it to spot predators before being eaten, locate prey efficiently, and navigate complex environments. The return is high, and the cost is justified. But remove the light entirely, and the return drops to zero, while the cost stays exactly the same. You are maintaining an expensive organ that produces nothing. In evolutionary terms, that is not a static situation. It is a slow drain on an organism's capacity to survive and reproduce, compounding with pressure across every single generation. The species that has taught researchers the most about this is a small, ordinary-looking fish called Astyanax mexicanus, the Mexican cavefish. What makes it scientifically remarkable is that it is not entirely a cave species. Surface-dwelling populations of the exact same fish live in rivers across northeastern Mexico with fully functional eyes, normal dark pigmentation, and standard visual behavior. Cave-dwelling populations of the same species live in underground aquifer systems nearby, in complete and permanent darkness, and are completely blind with pale, almost translucent skin. Researchers can study both groups, compare them directly, and track precisely what changed and what did not. This makes Astyanax the most informative natural experiment in vertebrate eye loss that science currently has access to, and the results are instructive in ways that go well beyond the eyes themselves. The cave populations did not simply lose eyes and stop there. The energy that would have gone into developing and maintaining eye tissue was measurably redirected into the lateral line system, the sensory organ that detects pressure changes and water movement at close range. This reallocation is not an inference. It is quantifiable in the tissue. Blind cave populations also develop significantly larger jaw muscles and carry a far denser distribution of taste buds than surface fish from the same species. Some studies have found up to 15 times more taste buds in cave populations, though the exact figure varies across different cave systems and populations. More taste buds mean better chemical detection of food dissolved in water. Stronger jaws mean more efficient processing once food is located. The organism did not simply lose something. It spent the savings on something else. So how does evolution actually remove an eye? Not through a single mutation, not through one catastrophic genetic event. The process is called regressive evolution, and it works through the gradual accumulation of mutations that degrade a structure that is no longer under selection pressure to stay intact. In Astyanax, research has traced eye degeneration back to the embryonic stage. The lens begins forming normally, following the same developmental sequence as in surface fish from the same species. Then it triggers its own programmed destruction, a process called apoptosis, within days of forming. The embryo starts building an eye and then, before the structure becomes functional, dismantles what it built. This is not a failure of development. It is development proceeding normally and then making a regulated, reproducible turn at a specific and consistent step in the sequence. The gene driving that turn is the sonic hedgehog gene. which is significantly upregulated in cave populations compared to surface populations of the same species. That single regulatory difference produces two downstream effects simultaneously. It suppresses eye development while accelerating expansion of the jaw and the mechanosensory structures. One upstream signal, two structural outcomes, moving in opposite directions at the same time. This is the detail that changes the framing most sharply. The cave fish is not losing its eyes through passive accumulation of mutations in a structure that selection no longer bothers to protect. The genetic pathway that removes the eye is the same one building the lateral line expansion. The demolition and the construction are running on the same signal. Evolution did not give up on eyes. It repurposed the budget allocated to them. The scale of convergence across species makes the point unavoidable. Eyelessness has evolved independently in at least 14 separate cave fish lineages around the world. Different continents, different fish families, no shared recent ancestor, but the same environmental pressure and the same evolutionary outcome. When the same solution appears that many times in unrelated lineages, it is not coincidence. It is the answer to a well-defined problem. The olm, Proteus anguinus, a cave salamander from the Balkans, illustrates how far this logic extends. It lives up to 100 years. It survives without food for up to a decade. Its eyes begin developing in the embryo and then regress before the animal matures. It navigates entirely through electroreception, magnetic field sensitivity, and chemical detection. It is what this evolutionary logic looks like after tens of millions of years of refinement. What the convergence tells you is that this is not a quirk of one lineage or one genetic accident in one isolated cave system. Evolution ran the same calculation in fish in Mexico, in salamanders in Slovenia, in crustaceans in caves across 4 continents, and arrived at the same answer every time. Remove the light permanently, sustain the pressure long enough, and the sensory investment shifts away from vision toward whatever actually works in that specific darkness. The exact mechanism varies. The SH pathway in Astyanax is not the same genetic route the olm took, but the destination is consistent enough that when researchers find a cave species with functional eyes, the working assumption is that it simply has not been isolated long enough yet. Give it enough generations, the math will catch up. Now here is where the story gets more complicated, because if your conclusion from everything above is that permanent darkness always produces eye loss, the deep ocean is going to push back on that directly. Deep-sea fish living below 1,000 meters often have enormous eyes, not vestigial remnants, not functionless tissue maintained by developmental inertia. Enormous, highly specialized eyes built for a specific purpose. The barrel-eye fish, Macropinna microstoma, has large fluid-filled tubular eyes that point straight upward, designed to detect any trace of light descending from the water column above. These are not accidents of anatomy. They are the result of strong sustained selection pressure for sensitivity in a specific direction. Why would a fish living in apparent permanent darkness evolve dramatically larger eyes rather than losing them entirely? The answer is bioluminescence. The deep ocean below 200 meters is populated by organisms that produce their own light. Firefly squid flash coordinated signals to communicate and attract mates. Lanternfish carry rows of small light-producing organs called photophores along their bodies. Anglerfish trail luminescent lures above their mouths to attract prey in the dark. Dinoflagellates illuminate in response to disturbance, sometimes lighting up entire wakes of ships at night. Estimates suggest that around 76% of deep-sea species produce bioluminescence at some depth, though the exact percentage is genuinely difficult to measure with precision. This is not ambient illumination in any meaningful sense. It is dim, localized, and unpredictable, but it is real light, generated continuously by the organisms around you. In that environment, the cost-benefit analysis for eyes stays solidly positive. What deep-sea fish do instead of losing eyes is rebuild them for the available signal. Deep-sea retinas shift almost entirely toward rod cells, the photoreceptors specialized for sensitivity in very low light, rather than color discrimination or sharp detail. Surface fish use a mixture of rods and cones calibrated for bright, color-rich environments. Deep-sea fish have retinas that prioritize detecting any photon that arrives, regardless of wavelength or direction. The giant squid has eyes up to 27 centimeters across, roughly the diameter of a dinner plate, the largest eyes of any animal alive. That is not an eye that gave up on light. That is an eye optimized to the physical limit for capturing it. The cave fish and the deep-sea fish are running the same underlying calculation, and arriving at opposite structural conclusions because their light environments are genuinely different from each other. So where does the deep sea actually abandon eyes? The answer is the Hadal Zone, the regions beginning at 6,000 meters and extending to 11,000 in the deep ocean trenches. At 10,000 meters, pressure reaches roughly 1,000 times the pressure at sea level. To make that physical, that is equivalent to 50 commercial passenger aircraft stacked on top of a single square centimeter of your skin. The Mariana Trench would swallow Mount Everest with more than 2 kilometers of water above the summit. In these zones, bioluminescence becomes genuinely sparse. The biological light that justifies large eyes in the mid-deep ocean is far less abundant here, and the food supply is so limited that caloric economy dominates every evolutionary trade-off. Every structure consuming energy without returning a survival benefit is a liability the organism cannot afford to maintain. The organisms that do survive in the Hadal Zone have converged on a small set of workable strategies. Some are scavengers built to detect and process organic material falling from far above, what marine biologists call marine snow, the slow drift of dead matter from shallower water that represents the primary food input at these depths. Others are predators of those scavengers. What almost none of them are is visual hunters. The sensory landscape of the Hadal Zone is chemical and mechanical, not optical. If you were designing an organism for this environment from scratch and were honest about the constraints, eyes would not make the list. They are expensive, they require light to function, and there is no light. The Hadal Zone is not a difficult place to have eyes. It is a place where having them is simply a mistake. The snailfish, Pseudoliparis sweri, was recovered from the Mariana Trench at a depth of 8,145 meters, making it one of the deepest fish ever directly observed and collected. It has eyes, but they are small and appear to be largely non-functional. It navigates primarily through its lateral line system and chemoreception. Put a cave fish and a Hadal snailfish next to each other, and you are looking at the same evolutionary logic applied in two different environments, arriving at the same structural outcome through different paths. The pressure, the cold, the food scarcity, and the near-total absence of bioluminescence combine to make the Hadal Zone the deep-sea equivalent of a sealed cave. The fish living below that depth increasingly reflect that calculation. The math on eyes stops working, and the biology follows. What the cave fish and the snailfish have in common, separated by thousands of kilometers and completely different evolutionary histories, is that both ended up in environments where the cost of vision became permanent and the return on that cost dropped to zero. The cave sealed off from sunlight, and the trench floor 11 kilometers below the ocean surface are ecologically very different places. But from the perspective of an eye trying to justify its metabolic budget, they are identical. No light means no signal. No signal means no function. No function means no return on a significant daily investment. The evolutionary response in both cases is the same because the problem in both cases is the same. What changes between them is only how long it took for the biology to fully reflect that calculation. Here is where everything you have understood up to this point needs one more piece to be complete. Blind cave fish still carry the genetic instructions for building eyes. Not fragments, not corrupted remnants degraded beyond usefulness across thousands of generations. The complete, intact blueprint is sitting in the genome of a fish that has been sightless for an enormous stretch of evolutionary time. The genes were not deleted. They were not scrambled by accumulated mutations to the point of being unreadable. They were silenced. That distinction is not a minor technical detail. It changes what the word lost actually means when we apply it to a biological capability. And it fundamentally changes the story of what evolution was doing the entire time we were watching it appear to remove eyes from these animals. In Astyanax mexicanus, the opsin genes, which encode the proteins that make photoreception physically possible, are present and intact in the genomes of completely blind cave populations. They are simply not being expressed. The regulatory machinery that would read those genes and produce functional proteins has been switched off, but the genes themselves are there, in every cell, in every generation, passed forward intact. When we say the cave fish lost its eyes, the intuitive image is deletion, that the instructions were removed and the capacity was permanently erased from the lineage. That is not what happened. The fish did not lose the blueprint. It stopped reading it while continuing to copy it forward. And a 2018 study from the Gross Lab at the University of Maryland, published in Current Biology, made the full implications of that distinction impossible to ignore. Cave-dwelling Astyanax were crossbred with surface-dwelling populations in the lab. The offspring developed partially functional eyes, not perfectly restored, but eyes that worked to a measurable degree. The genetic information for building eyes had been sitting, silent, inside those cave fish through thousands of generations of complete blindness, and it was still readable when given the right developmental context. The archive had not degraded. The blueprint had not become illegible. It had simply not been in production. Whether that remains true across millions of years rather than thousands is one of the genuinely open questions in evolutionary biology right now. We do not know where the threshold is. We do not fully understand what determines whether archived genetic information stays recoverable over evolutionary time or eventually degrades beyond use. That gap is itself interesting. What the crossbreeding experiment revealed is that the genome of a blind organism is not the same thing as the genome of an organism that never had eyes. The cave fish is not a simpler version of the surface fish. It is a different deployment of the same underlying instructions, running in a different configuration for a different environment. Researchers working on regressive evolution increasingly describe this not as loss, but as a shift in expression state, a distinction that sounds subtle until you realize what it means. The organism is, in some functional sense, carrying both versions of itself simultaneously, the seeing version and the non-seeing version. One currently expressed, one archived and dormant, both present in every cell of every individual in every generation. That is a genuinely strange thing to be, and it changes what the word organism actually means. This is the detail that reframes the entire story. Eyes were not abandoned, the cost-benefit analysis changed, and the expression of the genes that build eyes was switched off because maintaining that expression was more costly than the benefit it returned in permanent darkness. The same biological logic that builds eyes when light is available dismantles their expression when light disappears permanently. It is not subtraction, it is resource reallocation, and the archived instructions remain part of the organism's genome, not a byproduct discarded from the process. The organism did not discard the capacity to see. It stopped paying to deploy that capacity in an environment where deploying it returned nothing, while continuing to carry the full instructions forward through every generation, intact, at what appears to be a negligible additional metabolic cost. What that implies about living organisms in general is the part that tends to stay with you after the video ends. If evolution regularly archives capabilities instead of deleting them, then genomes are not only instructions for building what exists today, they are also records of what was built in previous environments, under previous pressures, and then set aside when conditions changed. The blind fish is not just a fish that cannot see. It is, in a precise molecular sense, one regulatory shift away from a version of itself that could. The gap between what it currently is and what it carries the instructions to become is not a question of missing information. The information is there, intact, and in at least some cases demonstrably still functional. What is missing is the environmental pressure that would make expressing it worthwhile. Think about what that means for a blind cave fish navigating its completely dark environment right now. It is reading water pressure through sensors along its body. It is detecting chemical gradients in the water to locate food. It is doing everything it needs to survive in permanent darkness, and doing it well enough that its lineage has persisted for tens of thousands of years without any vision at all. And inside every cell, there is a complete genetic blueprint for an eye it will never need and never build. That blueprint has been passed from parent to offspring across thousands of generations of total darkness. It has never been expressed toward its intended purpose. It has simply been carried, intact, through all of that time, a contingency for a future that may never arrive. The question of what else is sitting dormant inside living genomes right now, waiting for conditions that may or may not ever return, is one of the more productive directions evolutionary biology is actively exploring. If eyes can be silenced and retained across tens of thousands of generations in conditions that provide absolutely no selective reason to maintain them, the same logic almost certainly applies to other structures in other lineages across the entire tree of life. The answer to what those structures are is in the next video. Click on what is appearing on your screen right now. And if this one was worth your time, hit like before you go.